With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, so here the question is, study the experimental set given below and answer the following questions. And here the question is, name the chemical filled in the test tubes and explain how the above mentioned chemical would interfere with the experiment. So here in this diagram, as we can see, there are two setups. The first setup is labeled as A and the second setup is labeled as B. Now in the setup, it has a petri dish or a beaker and a test tube. The test tube is filled with a chemical and we have to tell what is the black colored chemical here. And both the setups has germinating seeds, but only the B setup has a gas liberated. There is a gas liberated. So here this experiment is done to demonstrate that germinating seeds can undergo anaerobic respiration. This experiment is done to demonstrate the germinating seeds can undergo anaerobic respiration. Anaerobic respiration means they can respire in the absence of oxygen and evolve carbon dioxide. So like any other organism, all parts of the higher plant organisms, they respire aerobically, which means with the help of oxygen. And in this aerobic respiration, the glucose molecules present in the cells, they get oxidized and carbon dioxide is released in this process. But certain cells of the plant parts, they use in abnormal conditions, they respire anaerobically temporarily for a certain period of time for their survival. And in anaerobic respiration, the cells of the plant parts, in the absence of oxygen, the glucose molecules are partially broken down to release the alcohol and the carbon dioxide molecules. So here this experiment is for to demonstrate that only. Now in this experiment, first take 8 to 10 healthy seeds, preferably bean seeds or pea seeds, and soak them in water for 1 day before using them in the experiment. So in the experiment, at first take 8 to 10 healthy seeds, 8 to 10 healthy seeds, and soak them in water. Soak them in water for a day before experiment. Now take 2 beakers or petri dishes and fill 3/4 of them with mercury. So then take 2 beakers or petri dishes and fill 3/4 with mercury. So this mercury liquid, and then after filling the 3/4 of the beakers, take 2 test tubes and fill them fully with the mercury. So these few germinating seeds, the 8 to 10 germinating seeds that we took, and peel the seed coats, peel off the seed coats of those seeds. Now these seeds should be carefully introduced into the inverted test tubes. The test tube is inverted here. The seeds are introduced in the inverted test tube of the, and this is done very carefully. So as the mercury is heavy in density than the seeds, as the mercury, it is heavier in density than the seeds. So what will happen is the seeds will be completely immersed in the mercury liquid, and they will rise to the top of the test tubes in both cases. Here we can see the germinating seeds are in the top of the test tubes in both cases. Now here the A setup, that is the control setup, and here in the A setup the seeds are boiled seeds, which means simply that they are dead seeds. So in the A setup the seeds are dead. The seeds are dead. But in the B setup, the seeds are alive. So after a few days, or after let's say 2 or 3 days, what we can see is that the B setup that had the live seeds, there is some gas. As we can see here, some blank space is here, and a gas will be collected at the top of the test tube for in the B setup, that is with the live seeds, live germinating seeds. So to find out what gas is collected, we will put potassium hydroxide. We will put potassium hydroxide in this test tube, and as the gas is carbon dioxide, it will be absorbed by the potassium hydroxide, and this place will be filled again by the seeds. So thus we can say that the gas liberated is carbon dioxide. So here the question says name the chemical filled in the test tubes. So the chemical that is filled in the test tubes, that is mercury, liquid. And how mercury interfere with the experiment? So mercury, as this is heavier in density than seeds, the seeds will rise to the top of the test tube and there is no oxygen in the setup, so the seeds will undergo anaerobic respiration and they will produce carbon dioxide. This will be our answer here.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.